also known as PCR. And what PCR does is it takes a strand of DNA found in the sample and it will amplify it to show up on an agarose gel. So we put our DNA samples into a thermocycler, which allows us to control the temperature of the sample at different intervals. And we heat it up to about 95 degrees Celsius so it would denature into a single-stranded DNA. We lowered the temperature so primers that we had previously added to the sample would anneal to the DNA at the different specific sites where we wanted them to attach. And then we raised the DNA, and the Taq polymerase that we had also added allowed the primers to extend to each strand of the DNA. And we repeated this process 35 times in order for the DNA to exponentially amplify the sites where the primers attached. And this allows us to see if these promoters are present in, or genes are present in our samples. We ran two different types of PCR reactions, a bead PCR and a single PCR reaction. The bead PCR came in a bead where all the materials were already in the epinorm tube and all we had to add was the DNA. The single PCR reaction, we had to add all the materials ourselves. And so we ran 5 different PCR reactions. We ran 2 bead PCRs for the invertase gene, which is found in corn, and the CAM-V35S promoter, which is found in almost all genetically modified corn products. And we ran 3 single PCRs for an NOS terminator, which again is commonly found in genetically modified corn products, and the EPSPS gene, which we know is, from previously said, is in the Roundup Ready, and the PAT gene, which is found in Liberty Link. So when we ran the IVR gel, we found that our positive control of corn showed the band that the corn gene was present, but only 2 of our 6 samples showed presence of this corn gene. This just could have been due to a lack of DNA, but the organic popcorn and the organic tortilla chips we knew found the corn gene. Then when we ran the CAM-V35S gel, our positive control, fluca corn, which we knew to be genetically modified, had the band present of genetic modification, and the organic tortilla chips had a thick band which showed that the organic tortilla chips were genetically modified, and the GMO-free tortilla chips showed a light band, but since we did not get the invertase gene from the GMO-free tortilla chips, we cannot be certain as to whether they were genetically modified. So when we ran our single PCR reactions, we ran it with 4 lanes. The invertase, which was our positive control, and then the NOS terminator, lane 2, the EPSPS gene, which is labeled 1, and the PAT gene, which is labeled 4. And for our popcorns, we found that the invertase worked, but the other lanes only showed some presence of shearing and no specific GMOs were found. With the tortilla chips, our organic tortilla chips we had found, which were genetically modified, did not have any specific genetic modifications present in our gels. And for the GMO-free tortilla chips, the positive control worked, and we had light